Welcome to IPMWORKS e-learning modules. My name is Eduardo Crisol. I work for COEXPHAL in Spain. This video is about module number 6, holistic IPM examples, and we will talk about the IPM of Tuta absoluta in tomato greenhouse crops. Tuta absoluta is a pest originally coming from Latin America, but currently it has widespread distribution worldwide. It's a top pest of tomato crops, and it causes severe damage in leaves and especially on fruits. Tuta absoluta receives a very high number of pesticide treatments, and this pest is resistant to the majority of the commercially available insecticides. Here, you can see a few examples of the literature explaining how this pest has developed resistance against some of the most common used insecticides. This is the reason why we need to change the way that we are dealing with this pest from a chemical control to a holistic integrated approach. So I will guide you briefly through 5 of the key elements of IPM of Tuta absoluta. The first and the second one are very much related. They deal with biological control. In the first case is augmentative biological control, which means the introduction of natural enemies. And the second one is conservation biological control, which means that we're going to be benefiting from the spontaneous species in the surroundings of our crop. And we will use strategies such as flowering strips to attract these beneficial insects into our crop and to boost their activity. The third element of IPM is mating disruption technique, using sex pheromones to reduce mating and thus population buildup. The fourth element is about using light traps to capture adults. And the fifth one is about using bioinsecticides and crop management to reduce damage. So, the first element, augmentative biological control, we have this picture, Nesidocoris tenuis, which is a very commonly used predator released in tomato greenhouse crops in southern Spain. And there are other similar species, other murids that are used in other European regions, such as Macrolophus. They all share the same characteristics, which is they are murids and they are generalist predators, which means that they're feeding from other pests, including whiteflies, thrips, and leaf miners. But they are also, for the purpose of this video, interestingly also feeding on Tuta absoluta eggs. The way that these natural enemies are released on the crop is when the plants are very young, they are in the nursery. So the Nesidocoris or the murids are released along with food additives. And this way we have a preventive strategy because we have already an established population of our murid bags in the plants. And this way we can prevent the population buildup of Tuta absoluta because they are going to be feeding on the eggs of the pest. We have another natural enemy that is commonly used in greenhouses, which is Trichorama achaeae. It's a parasitoid of Tuta absoluta eggs. In this case, we have a very small wasp. It's an enteroparasitoid, and the female is going to be laying eggs inside Tuta absoluta eggs. So you can imagine how small this wasp is. How to use this natural enemy is very easy. We are using these commercial carts that contains parasitized eggs with Trichorama adults. And so we are going to be placing between 50 and 100 of these carts for each hectare of our crop. And then we will repeat this treatment if it is necessary. We're moving into the second element of IPM. This is conservation, biological control. And for that, we need to talk about native species, Necremnus sutae. This species is widespread in Europe. According to the literature, this parasitoid wasp has adapted to parasitize larvae of Tuta absoluta. And we have some practical tips on how to use it and how to benefit our crops from its biology and from its action that we will see later on. These parasitic wasps, they are coming spontaneously into the tomato greenhouses from the surroundings. So they want to be reproducing on tuta absoluta larvae. And the thing is that this species is not commercially available, so it is important to learn how to recognize it so that we can keep it, we can secure its presence, that we can attract this species into our greenhouse. The way that the biology of this wasp operates is that the female wasp will be looking for galleries with living tuta absoluta larvae, and once it finds a suitable host, the wasp will paralyze the larva with venom, and then once the tuta absoluta larva is paralyzed, it will lay an egg next to it. As you can see, this is a gallery of tomato leaf, and then you can see in a circle the egg of the parasitoid next to the larva. Then a larva will emerge from that egg. Here we can see a newly emerged Necremnus sutae larva, and it's already feeding from its host. Of course, as the larva keeps feeding, it will grow in size. And sometimes there is occasionally you can find this situation where more than one egg is laid next to the host. Like in this case, we can see 3 Necremnus sutae larvae feeding on a tuta absoluta corpse. Once the larva finishes its development, it will turn into a pupa. And then from the pupa, eventually, there will be an adult emerging. Occasionally, there is a change in the behavior of this wasp. The female will start by paralyzing the larvae, but instead of laying an egg next to the larva, it will turn around and it will start feeding from the larva. So it will be feeding from the hemolymph. This way, the females are obtaining nutrients that they require to, particularly to produce eggs. So this is a very important behavior. And not just that, it's not just important for the physiology, but also for the functionality. Host feeding, we know empirically that is a key factor that boosts the rapid control of the pest. Sometimes in the field, we can observe the host feeding marks in the tuta larvae, and this is a very easy way to check that we have the presence of the parasitoid in our field, in our crop. We have carried out a field study between 2017 and 2019, where we have surveyed 64 tomato crop cycles in Almeria, in Spain. We wanted to check the biology of the parasitoid and how it did act controlling tuta absoluta under different circumstances. So here I am presenting you a couple of examples of what happened in the field. Here we're looking at data collected in a greenhouse in 2018. We can see in the horizontal line different sampling dates, and then on different colors we are looking at either in bluish colors parasitized larva or larva that was host fed, killed by host feeding. And then in reddish color and pinkish colors, we see a live larva of tuta absoluta. So in this case, we have a greenhouse where no insecticide was used. And you can see by looking at the numbers and the pattern that the percentage of parasitized larvae and host-fed larvae kept growing since the first sampling. And it reached a very interesting high number fairly quickly. We can actually say that we get a full control of tuta absoluta population in approximately 4 weeks, which is quite interesting in this case, again, without insecticide use. Now, to show you a different examples, we're going to see another greenhouse, a different one from the same year, where in this case, 2 insecticide treatments were used. These treatments they're represented by black arrows. And you can see a very similar pattern to what we've seen in the previous slide, but in this case, you can see that it's actually slowing. You see the dates on the horizontal line, and the population buildup of the parasitoid is actually growing slower. So it actually took approximately 8 weeks for Necrennus sutae to control the population of Tuta absoluta because there was a delay caused by insecticide use. So, very briefly, the main results that we found in this study is that there is a widespread distribution of the parasitoid of Necrennus sutae. We found it in 100% of all the greenhouses that we studied. In 70% of the occasions, the biological control was successful. It was quite fast, especially during the warmer months of the year. The key for this success was, first of all, to identify the parasitoid, to be able to recognize it. And secondly, very important, to avoid using insecticide use once the parasitoid is present in the greenhouse. Actually, in 30% of the occasions, the control failed because insecticide was used, and that caused a reduction on the parasitoid population. You can see more detailed information on the article that I'm giving you at the bottom of the page. It is very important to look for diversity, to look for flowering plants that have food sources. They also offer shelter and egg-laying sites for natural enemies, such as, in this case, the species Lobularia maritima that we can see in the picture. These flowering plants represent a very simple and yet very practical and solid strategy to reinforce conservation biological control. Actually, we know through a couple of studies in the literature that specifically the nectar of Lobularia maritima flowers can increase the longevity of Necrennus sutae female and also the number of eggs that are laid by these parasitic wasps. So not only will these flower strips contribute to conservation biological control of a number of pests, but specifically they will boost the biocontrol of Tuta absoluta. We're moving on to the next IPM element, which is mating disruption. In this case, we're talking about using sex pheromones that are going to be avoiding the mating of Tuta absoluta adults, and thus, of course, that will reduce the egg laying. It is very important to do this at the right timing. So it is essential when we are using the sex pheromones in a greenhouse to set them correctly before the plantation of the crop so that we will maximize their effect. To show you some results of the sex pheromones, in this case from a field study on a tomato crop in the field, you can see the different colors indicating either control or pheromone treated tomato fields. And in summary, we can say that the sex pheromone effect is pretty much causing first a delay of the population build-up and a reduced total abundance of Tuta absoluta. We are moving on to the next element of IPM, which is using light traps. In this case, we have a picture of a homemade light trap that pretty much this system is quite easy. It is a system that has two parts. One of them is a light that attracts Tuta absoluta adults. We're talking about moths, of course. And then we have a sticky surface underneath that can capture those moths. And so this is how it looks when it's in the dark. So it is important to say some practical considerations that we should be using light traps once we are start to see some of the Tuta absoluta moths in the crop. It is advised based on our empirical experience to use at least 10 light traps per hectare, and then turning on 2 to 3 hours during each sunrise and sunset time to maximize the captures of Tuta absoluta. Here you can see some data that we collected this year, and you can see, looking at the different colors, the number of moths captured per light trap per week in blue, and also you will see the percentage of fruit damage. It is very important to highlight that you can see the large numbers of Tuta absoluta adults captured throughout the whole crop cycle. And again, in this case, we have only one insecticide treatment that is indicated by that black arrow. But you can see that the average fruit damage is quite low and remains quite low, on average below 5% throughout all the crop cycle, which is very good results. We're moving on to the last IPM element, which is using bioinsecticides in crop management. In this case, we talk about Bacillus thuringiensis, which is a bacteria. This bacteria is producing toxins that are highly toxic to Tuta larvae once the larvae are consuming them, normally when they are sprayed over the crop. There is another integrated management that we can talk about, which is the manual defoliation of the plants, as you can see in the picture, especially when we are selecting those leaves that have galleries, as you can see on the top right. So this is a very simple manual way to reduce the abundance of the pest. And lastly, it is very important, especially when we have certain abundance, at least medium to large, I'll say high level of abundance of Tuta saluta at the end of the crop cycle. We will proceed with a solarization, which is using this plastic and water underneath, so that it will heat up and it will stay for at least 2 weeks, maintaining high temperature to eliminate all Tuta saluta pupae that can be found in the soil. And this is a very effective way to restart our crop and to make sure that we have no initial population of Tuta saluta present in our crop. So, all sum up, here we have the 5 key elements of the IPM, and I built up this table to show you how each of these IPM elements is actually focusing on a particular phenological stage of the pest. And you can see how some of them are targeting 1, 2, or the 3 of the different stages. So that is why it's essential to combine at least some, if not all, the IPM elements all together, so we can maximize their effects and try to reduce to the maximum the damage that Tuta saluta can cause to our tomato crops. And that was it. Thank you very much for your attention.